AS Level Biology 9700. Paper 2, Variant 2, October November 2024, question number 3 and 4, part 2. And question number 3 is, during transcription, base pairing occurs between nucleotides. Figure 3.1 is a diagram to show complementary base pairing between a DNA nucleotide and an RNA nucleotide. Only the base pair is shown in molecular detail. And here you can see that they have told us that the uh, explain why figure 3.1 does not include any phosphodiester bond. You can see they have shown one DNA nucleotide. So this side shows the DNA and this side shows the nucleotide of RNA strand. So, you know, the DNA contains the pentose sugar and this side shows the ribose sugar. So it means this is RNA and this is DNA. Now question is, include any phosphodiester bond. So students, the phosphodiester bonds are formed between two nucleotides. Nucleotide will join together with the help of a bond. Nucleotide. This it will be the phosphodiester bond, which will be formed between two nucleotide molecules in same chain, or strand. Okay, but here they have shown us two nucleotides from two different chains. That is one chain, one strand is DNA strand, the other strand is RNA strand. So here you can see that the phosphodiester bonds cannot be formed when there is DNA and RNA. They are joined together with the help of what? With the help of hydrogen bonds. So phosphodiester bonds has not been shown over here in this picture. Now, next question is, identify and describe the DNA RNA nucleotide pair shown in figure 3.1. You may add labels and annotations to figure 3.1 if you wish. Yes, I would like to explain this question. You can see the hydrogen bonds, which are non-covalent bonds. So they are formed between the DNA and RNA molecules. This is DNA molecule and this nucleotide has base pair, which is guanine. And on the in this side, this is cytosine. So guanine always pair with cytosine by making three hydrogen bonds. And here you can see three hydrogen bonds, which are very weak bonds. So they have been shown with the dotted line. So these are the very weak bonds which are formed between guanine and cytosine. So this is RNA and this is DNA. One nucleotide from DNA strand and one nucleotide from RNA strand. And on RNA side, there is cytosine. On DNA, guanine. They always pair in this way by making three triple bond or three bonds together. So here they are joined in this way. So figure 3.1 focuses on the hydrogen bonding between complementary bases. In a DNA RNA nucleotide pair, it's zoomed on two nitrogen base pair, that is guanine pairs with cytosine with three non-covalent hydrogen bonds, which are weak bonds. So pentose sugar is present on DNA side, already told you. in the previous question, that pentose sugar or deoxyribose sugar. Oxyribose sugar. The pentose sugar or deoxyribose sugar, both are same. So on the RNA side, you can see this ribose sugar is present. So it shows that this is the RNA molecule. And both of these nucleotides are joined together with these three non-covalent hydrogen bonds, which are very weak. So nucleotides in DNA strands are joined with phosphodiester bond, but here only one nucleotide from each strand has been already told you that nucleotide in the same strand, when joined together, then there is the formation of phosphodiester bond, or they join together with the help of phosphodiester bonds. But on the other side, here you can see the hydrogen bonds are there in this diagram. Now, question number 4. And question number 4 is, adult stem cells are undifferentiated cells that are found in most animal tissues. Adult stem cells can divide by mitosis throughout their lifespan to form identical stem cells, self-renewals, or to form cells that can differentiate into the functioning cells of the tissue. Mitosis is important for the repair of tissues. So explain what is meant by repair of tissue. First of all, mitosis, a cell division in which the chromosome number is maintained. Generation after generation. So this, and as a result of this mitosis, students, one cell is divided into two daughter cells. And parent cell has 46 number of chromosomes. The daughter cells will also have the same number of chromosomes. And further, they will divide in this way, but all they will have the 46 number of chromosomes. It means during this cell division, the chromosome number is maintained. And you can see this mitosis meant by repair of tissues. During this during the life process, and you very well know that sometimes the healing or tissue repair occur because tissue repair is the body's natural process of healing damaged tissues. So it involves a complex series of events that aim to restore the tissue structure and function as much as possible. So, the initial response to injury characterized by redness, swelling, the heat and pain, it helps to remove damaged cells and debris and to initiate the healing process. So cells in the surrounding tissues begin to divide and migrate to the injured site, replacing damaged or lost parts. So I would like to say that, so a new extracellular matrix is laid down, providing a scaffold for the new cells to grow on. So extracellular matrix is composed of proteins like collagen. So here I would like to say about this, that what happens when there is an injury. So a response to injury is characterized by swelling, heating, and pain. And the cells in the when injury occurs. you know, when there is a cut or an injury. So what happens? That is the cells in the surrounding tissues. Cells in surrounding tissue begin to divide and migrate. And migrate to the injured side, replacing damaged cell. So tissue repair is the body, I would like to say that this is a natural process. It has to occur, and healing damaged tissues, it involves a series of events that aim to restore the tissues, the structure and function as much as possible. So in this way, the tissue repair takes place by the mitosis. Now next question, the uncontrolled cell division is a characteristic feature of tumor formation differentiated from differentiated cell. Describe other features of tumor formation from a fully differentiated cell. So here you can see that about the question, tumor formation. And what is tumor? Tumor is actually formed by the cell division, uh, uncontrolled cell division. So uncontrolled cell division is a characteristic feature of tumor formation from any fully differentiated cell. Uncontrolled. You already told you in the previous question, one parent cell has to divide into two daughter cells, and those further divide into two daughter cells. So in this way, a controlled cell division takes place. That is, at the time of cell division, each parent cell will divide into two daughter cells. So this is the normal way. When there is uncontrolled cell division, in that case, you can say the tumor formation can take place. So normal cell division takes place in this way. It will be continued throughout the life. So we can say that but tumors arise from cells that have lost the normal control of cell division. Normally they are not dividing. As for example, the one parent cell will not divide into two daughter cells, it can divide into many cells. So uh, fully differentiated means the cell was once a mature specialized cell type. The implication is that even these specialized cells can revert to proliferative state under certain conditions. So we can say that in this way, the tumor formation, you can say expanding on the idea of uncontrolled cell division, and on that normal cells exhibit contact inhibition, meaning they stop dividing when they come into contact with other cells. So tumor cells lose this ability, continuing to divide and pile up, forming a mass. You can see they will divide, 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 and they will make a pile of masses. So one cell will not divide into two cells. So it will divide into many cells. So tumor cells lose their ability, continuity to divide and pile up, forming a mass. So in this way, they make uncontrolled cell division. So it is important to remember that while tumors can arise from differentiated cell, the process of tumor formation often involves differentiation and the accumulation of changes over time. If you want, you can choose few of these features and explain them in more detail. So I would like to say that this is enough for about tumor formation. Now I would uh, read this question, which I have already written for you. So normal cells undergo apoptosis when they are damaged or no longer required by the body. Notice they are, you can say. So next, uh, tumor cells can evade apoptosis, allowing them to survive and continue to grow. Tumor cells often exhibit genetic instability with an accumulation of mutations and chromosomal abnormalities, which contribute to uncontrolled growth and other malignant characteristics. So, in this way, here I have used this word apoptosis. Apoptosis is the normal planned death of damaged cells in your body. So, it is an important process that helps keep you healthy. But when it happens too often or not often enough, you can develop problems like tumors or cancer. So in this way, I would like to say that. So this is the natural process. So when they are damaged or no longer required by the body. So at that time, apoptosis takes place and those cells which are damaged or injured, so they have death over there. So tumor cells can evade apoptosis, allowing them to survive and continue to grow. So they will not let them to die and they will allowing them to survive and continue to grow. So tumor cells often exhibit genetic instability with an accumulation of mutations. So mutation can occur and chromosomal abnormality can occur. So all these things contribute to uncontrolled growth and other malignant characteristics. And you know, the malignant tumors can invade and destroy surrounding normal tissues. They can invade throughout the body. So, in this way, mutation can occur as a result of this mutation and chromosomal abnormalities. The tumors invade in throughout the body. So, in this way, they have uncontrolled growth and other malignant characteristics due to the mutations inside. Now, next question is telomeres. Next question is telomeres prevent loss of genes. Telomeres. Adult stem cells have chromosomes with long telomeres. Explain why long telomeres are an advantage to cells that carry out many cycles. You know the chromosomes. Chromosomes. Ends of each chromatid in a chromosome have telomeres. These ends are telomeres. So these telomeres are there to save the cell because uh, when cell replication takes place, after every replication, there can be the loss of DNA. So to avoid the loss of DNA, telomeres are there which act just like the caps. So after every replication, some part of the telomere is, you can see, lost, but DNA loss cannot take place. So it helps to avoid the prevents or it prevents the loss of the DNA. So we can say that the telomeres prevent loss of genes. So this is a fundamental concept. Telomeres are repetitive DNA sequences. So located at the ends of chromosome, they act as protective caps, preventing the loss of essential coding genes during DNA replication. So chromosomes with long telomeres, so we can see the piece of information stem cells which are responsible for replenishing tissues, undergo many cell divisions. So I would like to say the telomeres helps us in this way. So stem cells need to maintain ability to divide over and over to replenish tissues. So in this way, I would like to say the stem cells which have ability to divide, so these stem cells need to maintain ability. So their ability to divide should be maintained. So the long telomeres provide a buffer allowing stem cells to undergo many rounds of replication. As cell has to divide again and again, it has to replicate again and again. But after every replication, there is loss of ends of the chromosomes. So there is no loss of DNA, only the loss of telomere takes place at that time. So allowing stem cells to undergo many rounds of replication before reaching the critical telomere length that it triggers the uh, sensine apoptosis. So long telomeres maintain the stability and integrity of the chromosome. Shorter telomeres are associated with an increased risk of DNA damage. DNA damage. So this is T, not G. And chromosomal abnormalities which can lead to cellular dysfunction or even cancer. So in this way, I would like to say the telomere save us by many ways because it maintains the stability and integrity of the chromosome. Otherwise, the DNA can be lost during each replication. So here you can see over here, our next question is hematopoietic stem cells. So hematopoietic stem cells are adult cells that are located in the bone marrow of bones. HSCs have a role in the formation of blood cells. So here you can see the figure 4.1 is an outline summary showing the formation of some of the different types of blood cells that can be formed uh, from HSC. The first stage is the division of HSCs to produce progenitor cells. These cells are also able to divide by mitosis but are not stem cells. And key progenitor cells, they have been shown with a dark color. So HSCs, they divide into HIC, self-renewal, and CLP cells and CMP cells. So CMP cells, MEP cells, GMP cells, which are further divide, MEP divides into immature blood cells, and these immature cells will further change into mature red blood cells. So mega karyoids जो हैं they will into platelets, they will produce the platelets or GMP cells, they will immature neutrophils and immature monocytes, they will change into monocytes or mature neutrophils. On the other hand, CLP cells, pre beta cells and pre-T cells, immature beta lymphocytes or immature T lymphocytes. So mature beta lymphocytes and mature lymphocytes helper and T killer cells or and a immune response will be there. So here they have shown this summary in the form of this diagram. Now question is with reference to figure 4.1. Explain why GMP cells, which are progenitor cells, cannot be described as hematopoietic stem cells. Progenitor cells, hematopoietic cells. So they will not be considered as hematopoietic cells. So here we will move towards the question, that is the with reference to figure 4.1 GMP cells. So here I would like to discuss about these cells, that what happens, why they are Well, here you can see that HSCs possesses a unique ability to self-renew, meaning they can divide and create more HSC cells, ensuring a constant supply of stem cells throughout the life. So the diagram HSC shows the dividing to produce more HSC self-renewal cells. Here you can see the self-renewal cells in the diagram. Let me move back to the diagram. Yes, you can see over here the self-renewal cells, which can be formed by. On the other hand, the diagram, on the other hand, the GMP cells differentiate into specific cell types. As for example, you can see the neutrophils and monocytes. So these are the specific cells which are differentiated. So HSC are pluripotent or multipotent, you can say, meaning they can differentiate into all the different types of cells. The diagram shows HSC giving rise to common myeloid progenitors. Myeloid progenitors, and you can see the which subsequently branch into all the various blood groups lineages. So CM more restricted to their differential potential. So they are committed to becoming either granulocytes or monocytes. You can see over there. So they are lost the ability to form other blood cells type like lymphocytes or red blood cell cannot. So we can say that GMP cannot be HSC. So they have limited differentiation potential. They are already committed to specific lineages and cannot give rise to all the different blood cells like HSCs cannot present. So in this way, I would like to say that GMP cannot be because they lack self-renewal capacity. Already told you in explanation that they have the ability to self-renew. Already shown you in picture. So whereas they cannot. Once a GMP differentiate, it cannot replenish the HSC pool. So they have limited differentiation potential because they have limited differentiation potential because they already committed to specific lineages and cannot give rise to all the different blood cells like HSC can. HSC has variety, self-renewal, and neutrophils, monocytes, all different types of cells are more specialized, representing a latent stage in the process committed to producing specific type of cells and lacking the stem cells properties of self-renewal and broad differentiation. So in this way, both of they are different. That is, one of them have ability to self-renewal and the other have the ability, they don't have ability to self-renewal, but they have ability to differentiate into specific type of cells. So in this way, they are not cannot be described as hematopoietic stem cells. Next question is figure 4.1 shows that monocytes differentiate into cells type X, which has a similar function to neutrophils, names types X. So this is X monocytes, this is X, and this is Y. So first of all, we have to talk about this and we have to talk about this. So what are these X cells? They are macrophages. They are macrophages, or we can say phagocytes or mature monocytes. So in this way, we can say that. And what is this Y? Y mature beta lymphocytes. So they will secrete the antibodies. as an immune response. So why are the antibodies? Next question, we macrophages and antibodies. So the differentiation of T lymphocytes begins in the bone marrow and continue in an organ known as the thymus to produce fully differentiated T helper and T killer cells. Thymus glands are present in the beside the chest bone. Their size is of T size, so they are very small in size, so they are present over there. So what happens? The T lymphocytes begins in the bone marrow. They are produced in the bone marrow and then continues in an organ. Then their continuity of life takes place in the thymus. So the question is, why T lymphocytes that bind to self antigens belonging to the body's own need to be destroyed in the thymus? So T cells are trained in the thymus, are generated in the bone marrow, but mature in the thymus. So during this maturation process, they learn to distinguish between self and non-self. So the immune system must not attack the body's own tissues. This is known as self-tolerance. If T are allowed to recognize and attack self-antigens, they would cause autoimmune diseases, where the body's immune system mistakenly target in its own cells and tissues. So T cells are in the thymus, the T cells are present with self antigens. So the T cells have uh, these with self antigens to strongly bind to these self antigens are identified as potentially dangerous and are eliminated through a process called negative selection. So in this way, we can say that after this explanation, I would like to read the self antigens are described. So explanation can be uh, T cells are generated in the bone marrow. It is they have told us that they produce in bone marrow but mature in the thymus. So during the maturation process, they learn to distinguish between self and non-self. Self and non-self, they know which are the body cells and which are the aliens or which are the antigens which are not the part of our body cells. So self and non-self, they distinguish between them. They know very well. So the immature, so the immune system cell does not attack the body's own tissues. Our body is not attacked by our own immune system. So this is known as self-tolerance in the thymus. We can bear ourselves. So this is a type of self-tolerant process. So the immune system must not attack the bodies. This is known as self. In the thymus, T cells are presented with self antigens. T cells that strongly bind to these self antigens are identified as potentially dangerous. So what happens? These strongly bind with self antigens and are eliminated through a process. Then they eliminate them by negative selection. So this ensures that the mature T cells population largely devoid of cells that would attack self. So in this way, I would like to say there is no formation of the cells which attack on over their own body cells or on cells. So it was all about question number 3 and 4. Please like, share, and subscribe my channel. Thanks for watching.